when people ask me what might be my greatest contribution, it's the mentorship to the students that I have been in Bloomington. I love being there. I love being a teacher. I love the intimacy of working with young people. And when I look at where these individuals are now functioning at the Lillys, the Novos, the Mercks, the J&Js, and in academia, that's the next legacy, right? You want to be transformative? Plant people.
Welcome to Off the Record, a podcast featuring leaders on IBJ Media's Indiana 250 list. I'm Nate Feltman, CEO of IBJ Media, which publishes the Indiana 250, a list of the most influential business people in the state of Indiana. Today, I'm joined by scientist and entrepreneur Dr. Richard DiMarchi, a distinguished professor of chemistry and the Gill Chair in Biomolecular Sciences at Indiana University. Richard DiMarchi's research helped lay the foundation for the new class of obesity drugs that are giving people hope and transforming lives worldwide. Richard grew up a Brooklyn native and received his bachelor's degree from Florida Atlantic University before arriving in Bloomington, where he got his PhD in biochemistry from Indiana University. He went on to do his postdoc fellowship at Rockefeller University under the mentorship of Nobel laureate Bruce Merrifield. Dr. DiMarchi started his career at Eli Lilly and Company as a research scientist. Over the course of a 22-year career at Lilly Research Labs, he focused mostly on diabetes research. While at Lilly, he discovered the active peptide ingredient in Humalog, an injectable, fast-acting insulin drug to treat diabetes. Dr. DiMarchi retired from Lilly in 2003 at the age of 50 and then launched an incredible entrepreneurial career where you would go on to great success with a number of new companies that you launched, and then joined the chemistry faculty at IU Bloomington. Since retirement from Lilly, Dr. DiMarchi has co-founded 6, but now I understand maybe even more, biotech companies. And earlier this year, the American Association for the Advancement of Science presented Dr. DiMarchi with the Manny Baumick Breakthrough of the Year Award for his GLP-1 research. And Dr. DiMarchi has won numerous other awards, of course, including being inducted in the National Inventors Hall of Fame in 2014 for your work in the development of many medicines over many years. Dr. DiMarchi is a co-inventor on more than 100 U.S. patents and has co-authored more than 250 peer-reviewed scientific publications. His latest venture, MBX Biosciences, is developing treatments for endocrine and metabolic disorders. The Carmel-based company recently went public, raising almost $165 million in its initial public offering. Here's our conversation. Dr. DiMarchi, welcome to Off the Record podcast. Thank you for making time to be with us today.
Nate, thank you so much for the invitation and the very, very kind comments. It's really a pleasure to be here with you.
We appreciate it. And there's a lot of science to get to it. And it'll be wonderful to kind of hear from you, kind of a layperson's description of some of the things that you've been spending your life on. But I want to talk first a little bit about your early years. You grew up in Brooklyn, went to school in Florida, and then moved to Bloomington for your PhD work. When did it all come to you that you wanted to spend your life or spend time as a research scientist?
It's a great question. And 2 weeks, I'll turn 72, and I've been 50 years in the state of Indiana, coming here as a graduate student in August of 1974, and it all passes so quickly. The time in New York was a fundamental importance. You know, New York is just a special place, and the decade of the 60s was just filled with change: women's rights, civil rights, and the trip to the moon, right? Who wasn't touched at that age in 1962 when JFK said, we're going to the moon, if you remember, not because it's easy, because it's hard. And those who had mathematical, quantitative capabilities just sort of ran and grabbed their slide rules. No calculators, no computers at access to say we're going to the moon. Nobody ever asked why we were going to the moon, but you knew it wasn't just that trip. It was what would come after it. And in that period of time, in the 60s, we saw the advancement of the molecular basis of life, right? Watson and Crick, Jim Watson, a graduate student in Bloomington, working for another Nobel laureate, Salvatore Luria. 1953, the structure of DNA, and then from there, all that has occurred. We went to Florida because my dad died at a young age. My mother had remarried. We became state residents in Florida. I had a scholarship to go to this school that few people knew of. But now, because of basketball, folks become familiar with it. And in the course of being there, the chairman of Indiana University Chemistry, Gene Cordes, came to visit. He was one of two co-authors of the textbook we used to teach biochemistry and convinced me that the protein chemistry in Bloomington was as good as anywhere in the world. And I believe that was correct. And that's what brought me to the state of Indiana. So these universities, Purdue, IU, Notre Dame, they are magnets for recruiting talent to the state of Indiana, some of which remain and some of which make important contributions to the science and to the economy of the state.
So as a young student, it was the events of the day. Was there family that had to spend time in medicine or research that drew you? Or was it something that hit you while you were in school or down in Florida in college that said, research is something I'm really interested in?
We tend to do what we're most capable in doing, right? And so the quantitative sciences were always an attraction to me. I didn't take to the linguistics, can't speak a foreign language. I don't feel as if I have any artistic talent in drawing, sculpting, or the like. And as I drew closer and closer into the physical science, there was a certain attraction to chemistry, and particularly organic chemistry as applied to large molecules, peptides, proteins that have been such a big part of my career.
So you finish your PhD at Indiana, and then you get an offer to join Eli Lilly soon thereafter, I understand. And you're a research scientist focusing, of course, Lilly is famous for diabetes research and work, and that's an area that you're spending time in. And this is where you focus on proteins and peptides, and that begins to take shape. And that's the genesis of the weight loss drugs that we know today. Do I have that somewhat right?
A half century, 50-year journey. And my interest in these large molecules was at a time when we chemically could not prepare them. So you were either extracting them from blood, from urine, from organs, and that is just not a commercial business. Insulin was sort of an exception because of the life-altering properties of being deficient in insulin. But the pharmaceutical industry is a chemicals industry, right? We make chemicals. We sell chemicals. They're made value because of their biological content. But at the end of the day, these are things that we describe in specific molecular terms. You know the molecular mass of that drug, whether it's aspirin, penicillin, Prozac, right, fluoxetine. And these large molecules we were incapable of making. I went to New York from Bloomington, never thinking I was coming back, to work with a fellow who had advanced legendary synthetic capabilities for making peptides. At the point that I got there, they were relatively small peptides, the synthesis of something like the active ingredient in these anti-obesity peptides. You mentioned GLP-1, GIP is another one of those ingredients, was quite frankly beyond our capabilities at any scale. While I was working in New York, what emerges is what has become the biotechnology industry. Right? And so in the early 70s, these were enzymologists and microbiologists that found that you can manipulate microorganisms to make these complex proteins. I mean, one of the more colorful proteins was follicle stimulating hormone, which is used for facilitating fertility. That hormone had been called holy water. It had been extracted from the urine of nuns, with the Catholic Church having a significant ownership in the company, Serono,
is that right?
that was producing this. 10 days, 10 nuns was enough for one treatment of an individual trying to promote fertility. Holy water, right? All of that has disappeared over the decades as we now use microorganisms. So my synthetic capabilities that I had initiated in Bloomington, elaborated in a laboratory where the mentor, Bruce Merrifield, went on to win the Nobel Prize in '84, put me in a position as Lilly was beginning to produce human insulin in these microorganisms, bacteria, to make a contribution. And when I was recruited, I came to learn the technology. I wasn't looking to come back to Indiana, with all due respect. I wasn't looking to work on human insulin. I wanted to learn the chemistry of making these proteins in microorganisms so that I can then go on and do some other things.
Of course, today we have these household names. Lilly has Mounjaro and Zepbound, and we hear Ozempic and others that are now. What we see today in these weight loss drugs, the genesis was decades ago. Is it because of what you just described, the development of the biotech industry, that ultimately led to this? Or was there some missing opportunity decades ago, but it took us years to prove out that these drugs could be safe? Talk about that so we can understand that a little more.
There were at least three levels at which we had confronted obstacles to achieving what we have achieved. The first and fundamental was obesity was not considered a disease. This was sort of a lifestyle condition. If you could just control your diet, you wouldn't have this problem. So why are we developing drugs for this condition that is less than a disease without a clear regulatory path for how we would do that? Secondly, was just the belief that you wouldn't be able to do this safely, that you couldn't get a profound enough decrease in body weight, maybe a few pounds, maybe as much as 5% reduction in weight, but not the sort of spectacular efficacy that we're seeing today. And the last, as folks would tell me, as if I didn't get the first 2 points, was even if we could register it, even if we could get a greater effect, folks would not be willing to inject themselves to achieve this effect. I've always been a great believer. If you have a life-altering disease, you will stand on your head and inject in your eye to get what you need. And obviously, the advances in the technology, these injection devices, the fine needles, once a week administration has made it far easier. But what drives people's interest is the fact that they're losing 20, 25% body weight, which is truly spectacular, well beyond what anyone was anticipating until it was achieved.
When you look forward over the next couple of decades, and as you mentioned, now obesity is, of course, recognized as a disease. But when you look forward and you think about what the implications are, in Indiana, we talk constantly about the high cost of healthcare. We know, obviously, there's public health outcomes. There's bad public health outcomes in Indiana and a lot of other places, but we talk about it being really a problem here based on a lot of factors, obesity being one of them, but many others. Do you see that there is an opportunity here to drastically improve health outcomes and decrease the cost of healthcare through the use of these types of drugs, or is that not that kind of opportunity?
That's the vision. That's my hope. You inserted the economic issue of decreasing cost of healthcare. That is a bigger issue than just the technology. But I can tell you, in the late 80s and early 90s, during my time at Lilly, I was the one who introduced the company and promoted the work that was being done on these so-called incretins. These are peptides that are produced in your gut that are signaling to the pancreas that you're taking nourishment, and therefore one has to promote insulin biosynthesis and its release to deal with this fuel substrate that you're taking on. Additionally, it's slowing gut motility and signaling to the brain, gut-brain communication, that you're now satiated, you should not continue eating. That's the physiological function of these substances. But at that point in time, as I shared with you, obesity wasn't a disease. There was a sense you could never do what we achieved. The cool place to be playing was cancer and CNS and maybe inflammatory diseases. Diabetes, as important as it was, and metabolism in general, was dated. It was backwater. And here we are now, where we look at what has come from the investment in diabetes, and we should talk about that a bit, because it was diabetes that drove the investment to get to obesity. In many ways, how ironic as it is, type 2 diabetes was the incentive that allowed us to get to these spectacular drugs. That's where Novo was functioning. That's where we at Lilly were functioning. And here now we see it's not just diabetes. It's not just obesity. Sleep apnea, possibly osteoarthritis, kidney disease, liver fibrosis, addictive behaviors, and maybe even cognition. Well, guess what? Metabolism now is neuroscience. Metabolism is endocrine-based carcinomas. And so I like to think that we're working at a higher level where this is not just obesity. This is human health, and particularly human health in the aged.
And even cancer, right?
Endocrine-based cancers are certainly in play.
So do you think we will see, of course, there'll be studies and studies and studies on impact of these drugs on all the things you just mentioned. So do you think that there's a possibility in the next 5 years even, or 10 years, as the studies are completed and we see improvements in all these different, potentially, these areas of human health, that this could expand into treating lots of different, not just obesity, but maybe particular, to prevent other types of disease. Is that a possibility? Am I thinking of that right?
Oh, absolutely. I think for those who are looking more closely and beyond these spectacular effects to lower body weight, to control diabetes without the risk of hypoglycemia, to decrease cardiovascular outcomes, is how did we get here? And what might these be telling us in how do we get to other drugs that might be operating at this higher level that I commented upon that could have broad systemic ability to improve human health. And if there's a secret within these molecules, it is this ability to recruit multiple mechanisms of action. GLP-1 is sort of the first stage. It ignites your ability to resensitize other metabolic hormones. That is why GIP, which is the second active ingredient in Mounjaro, right, in Zepbound, becomes capable of doing what it's doing. GLP sets the stage, much like the first stage of a rocket, that then leads to the ability of other substances to do the good things that it needs to do. The third that Lilly has integrated in a drug they discovered, right, retatrutide that is being developed, is glucagon. And glucagon is something we embraced in Bloomington that was incredibly iconoclastic. Glucagon had been viewed as something that had to be inhibited or blocked for treating diabetes. And here we were using it to treat diabetes. And so it is this integration of multiple substances that gives us the spectacular efficacy. I had someone come up to a rather respected person and said, so what's so spectacular about 1 + 1 + 1 = 3? Meaning the three ingredients in what would be in retatrutide right now. The point of the matter, it's 1 - 1 + 0 = 3, because while GLP was showing efficacy, glucagon was supposed to go in the other direction, and GIP was believed to be inactive in pathological states. And so what we're doing is replicating physiology in the synergy, this concert of active entities that keep us healthy in pharmacology at a much more complex level. Can we reproduce that in other disease states? How do you, as a discovery scientist, embrace this higher level of complexity to find such drugs? Because we have a way of reducing the complexity to something that we can deal with. And in having single mechanisms, we're getting greater safety, but we're losing the efficacy that you can otherwise get if you can tackle this at a higher level. So that's the excitement of the age.
We're living in exciting times, clearly.
Oh, it's the end of the beginning, right? And that is what's so exciting, because what comes next, I think, is doubly exciting.
I mentioned in the intro that you worked under a Nobel laureate by the name of Bruce Merrifield. Can you just talk a little bit about how much he influenced your career? And I know you're having similar impact on thousands of students that have come through your laboratories in Bloomington. Maybe just talk a little bit about the importance of mentorship and maybe your experience there, and maybe some of your experiences in Bloomington, working with the students that have come through your lab and have trained with you.
Robert Bruce Merrifield, but Bruce Merrifield was of fundamental importance to me. A very different character than anyone I had bumped into. You grow up in Brooklyn, New York with a name that ends in a vowel. You're thinking DiMaggio, you're thinking Pacino, you're thinking De Niro. You're not thinking Tony Fauci, okay? right? And yet Bruce was someone who sort of stepped out of a Steinbeck novel, The Grapes of Wrath, and the family moving from Texas to Bakersfield, California, educated in a community college, finished at UCLA, came to Rockefeller in 49, and just had this sort of stoic intensity that he wanted to find a way to make these peptides, and developed a technology that was a completely orthogonal to what anybody else was doing, right? To actually do synthesis on a solid support. It just didn't make sense. And not to characterize all of the intermediates. When he submitted his landmark paper in 1962, it was published. There was one reviewer that has now come to know that says this work is not only unsuitable for publication, but should be suppressed because it is corroding the way we go about chemical synthesis. He was transformative. And it just demonstrates that you don't only have to be a visionary, you have to be incredibly tough to continue on, because that kind of criticism is devastating to people who don't have that West Texas toughness that he had. And so when people ask me what might be my greatest contribution, it's the mentorship to the students that I have been in Bloomington. I love being there. I love being a teacher. I love the intimacy of working with young people. And when I look at where these individuals are now functioning at the Lillys, the Novos, the Mercks, the J&Js, and in academia, that's the next legacy, right? You want to be transformative? Plant people.
Let's take a quick break. This is Off the Record Podcast.
Get caught up on the state's top business news every business day with the Inside Indiana Business Radio On Demand podcast. Available now at InsideIndianaBusiness.com, Apple Podcasts, Spotify, or wherever you get your podcasts.
As we've discussed, you start out as a scientist, both in school and Lilly, but in your second act, starting at age 50, really, so young. I know 50 is very young. I will attest to that. You turn into an incredibly successful entrepreneur, creating companies that then sold to the likes of Roche and Novo Nordisk. But talk about how a research scientist makes a transition from scientist to entrepreneur, and what that transition and mentality looks like. And I know you had partners in all of these ventures, but talk a little bit about. Not all researchers become entrepreneurs or, work to create new companies, which you've done many times. But maybe talk a little bit about that.
You're right. I was in mid-career. I'm sure it was driven by my impatience. I'm sure it was driven by my vision that these large molecules could be the medicines that they've proven to be. And that's not just these peptides as anti-obesity agents. It's proteins for treating inflammatory diseases. It's the RNAs that have been used as vaccines for COVID. I was just a believer that these large molecules would constitute miraculous drug substances. And the fact that they had to be injected, parenteral products, was, as I said, something that could be accommodated if the pharmacology was miraculous, right? That it was that much different than the traditional oral drugs that we have been about. And so that's what led me to a point where I felt Lilly's focus on neuroscience at that point in time, understandably, the successes of Prozac, Zyprexa, Strattera, Cymbalta, would tell you that there would just be a stream of that innovation coming, and the things I want to do became backwater. And therefore, I said, if that's the case, I'd rather flee than fight. You just get exhausted with that. And the exit was to start a company with a good friend, Pete Schultz, who, much like my reason for coming to Indiana to learn biotechnology, had this academic capability of making proteins with non-native amino acids. Nature is restricted in the building blocks it has. His technology was capable of introducing those very useful chemistries that medicinal scientists needed. And so we set this up on the West Coast. They were willing to do it in Indiana, interestingly enough. And I chose to do it on the West Coast, not because I wanted to leave Indiana. It's because I believed the capabilities and the track record was resident in La Jolla, and I would go to California and learn it and replicate it back here in Indiana. And it worked to a T. Simultaneously, I started my academic position. Gus Watanabe was of instrumental importance, David Klemmer, in saying, boy, if you're interested in doing academics, why don't you stay in Indiana? Because I had a faculty position offered to me at the Scripps Institute right across from where we were setting up the company. So 1 week in California, 1 week in Indiana for at least a year. It was a crazy life. And I can tell you, having been head of biotechnology at Lilly, when you arrive at a lab on an academic campus and there's no one there, there's no equipment there, you really do begin to wonder whether you were insane in trying to do what we've been able to do.
As I mentioned, you've had business partners, and you just mentioned one of them in one of your earlier ventures. And many people talk about how founders of companies aren't necessarily the people who are going to take the company to the next level and ultimately have success. But can you talk a little bit about, you've had more than one business partner, I know, in different ventures. How do you go about choosing a business partner, and what makes for you, and what has made a good business partner with some of these ventures?
It's a great point. And again, the reason why I went to California, because I wasn't seeing folks here in Indiana who had that experience and capability. And you learn the rules of the road in working with the venture community, and how do you not only deliver breakthrough science, but generate a profit in doing this, because if there isn't a profit, there won't be willingness to reinvest. Fritz French was of utmost importance in getting Marcadius started. And when you ask how you select such a person, I want someone who complements what I do and with the toughness to tell me what I need to know that I may not want to know, to restrict me in what I'm doing so that we get focused on the things that have to get done in a timely fashion. And Fritz was just a phenomenal leader of Marcadia, incredibly analytical, integrity that is impeccable. And so it's sort of the left brain, right brain phenomena that if you're going to have people like myself who are operating or even living outside the box, you really need folks that keep the organization grounded. From there, Kent Harlick, who then was a junior member, helped me form MB2 as we got technology back from Roche that Novo then acquired and now started MBX, and he deserves the lion's share of the credit for getting it to this public status. And Derek Small was incredibly important in helping Adam Zlotnick and myself take Adam's antiviral technology out of IU into assembly as a public entity. And so what you now see is in Indiana, we have that expertise that drove me 20 years ago to California. That's the most important contribution, because without that, Christian Sherman, right, doing what she's doing, Tim Knickerbocker. I mean, these are really important people.
The people are important. The funding is also important. And I would assume if you look back 20 years ago when you made that California decision, also funding had to be part of it, because often people will say, well, we're flyover territory. Has that changed in the last 20 years? Does it matter now? Can you be based in Indiana and raise the dollars as long as you've got something?
I think you can. 5 a.m. was, the very name tells you they get up early in the morning and they're funding early initiatives. John Diekman became a very good and trusted friend because he was funding Ambrex, that first company we started in California, and then Marcadia. It was Gus Watanabe who had seen the science we were developing in Bloomington and said, we must start a company in Indiana. And I said to him, I'm not opposed to that, but Gus, I've got a company in California. I got an academic activity. How much more can we do? And he said, we're going to do it. All right. And sure enough, we put it together and it was quite a success. But you're right. The VC community has to make a profit. And once they make a profit, you're more likely to have them return. So folks without a track record of the type that I have will still struggle. But The fact that we have shown that the geography can work, and the fact that you can connect with people who can make introductions and vouch for you, makes all the difference in the world.
In economic development circles, we constantly talk about how can we create more Richard DiMarchi success stories like you've had, and how do we take research great research that's being done at all of our universities and turn them into the grand scale, the next Lilly, but like the next MBX. I'm curious from your perspective, are there steps that you think we should be taking as a state, or what universities can be doing to further encourage researchers like yourself to turn their research into practical applications, and most importantly, from an economic development standpoint, to turn them into new companies that'll hire new employees and do wonderful things? As a state and as a university, what more can we be doing?
That is such a large issue, and why I stay associated with BioCrossroads, and why I'm so pleased with the things that I see that are happening on the Purdue campus and the IU campus. While I'm on IU faculty, and I have great pride in what we're doing in Bloomington, I stand for the success of Central Indiana and Purdue for as much as I do for Indiana. The work that Phil Lau is doing, Pete Kissinger has done, Graham Cooks has done. I take great pleasure in seeing them succeed. We're far too small to think in a zero-sum world of competing against one another. I think Purdue historically has been more inclined as an engineering school to think about translational contributions. I think IU has been slower. There were those when I was recruited to Bloomington who wondered, why are we recruiting this sort of retired executive? What does he have left to contribute? And I think President Whitten and Russ Mumford are really trying to promote the sorts of things that I have been about. I think we've just been a little too provincial in focusing on what academics define, with not enough emphasis on the translation. It doesn't mean doing it at the expense of that pure academic advancement, but as a complement that just makes us all the smarter. When people ask me what was special about my own training, I just sort of indicate that I entered into Lilly in product development and working on human insulin at the back end. And seeing what was happening at the back end gave me a huge understanding of what do I have to do on the front end to be able to market it at the back end. I'm fearful that those in academics don't have sufficient exposure and understanding. And as a result, they just retreat back into what they have done, which is not to translate, right? To be dependent on a public funding, RO1s, NSF type grants. This is all possible. Adam Zlotnick showed that in Assembly. We've shown it in a few companies, and I'm here to help.
Is it recruitment by universities? If a university says, we want to create this type of opportunity, is it recruitment of the researchers and professors that is key? Is it something different where it's convincing or incentivizing existing researchers to think about pairing up with an entrepreneur and thinking about translating their research? Is it both?
Recruitment, retention, and setting a culture that says we will be more diverse than we were before. It's not that we dislike what we were doing. It's just that we want more. I think we should celebrate diversity in multiple forms, and this is one of them. It is good for the state of Indiana that we have made these economic contributions. When I look at a number of the people that I've brought to the state that were associated with Novo, that are now at the IBRI, who have transitioned to Lilly, that are working with me on these additional startups. When I look at the revenue that flowed in the state from these purchases made by foreign enterprises, be it Novo in Denmark, be it Roche in Switzerland, that's good. And so we want to do more of this, not at the expense of the traditional excellence, but as a complement. And one last point, I've often said that the growth in places like the Bay Area were folks who came out of successful biotech companies. Hybridtech in San Diego, with its success, was a pool, provided a pool of individuals. So when I look at Alan Palkowitz over at the IBRI and how spectacularly he is functioning in his second career after Lilly, I've said there have to be other individuals like this that might be better for the ecosystem if they were doing what I was doing, what Alan was doing, without corrupting or undermining the ability of the Lillys, the Cooks, the Roches to do what we want them to do.
So we talked about MBX a little bit, but just had a wonderful success story in terms of the initial public offering. Describe the primary product or products that MBX is focused on today. And I'm assuming, but I don't want to assume, that the part of that success is the wonderful reception these obesity drugs are getting, and therefore MBX had a wonderful reception as in that tailwind. But is that partially correct?
So MBX is something that Tim Knickerbocker, Kent Harlick, and I dreamed up. You just go in the kitchen and you just whip up a souffle, and it starts with what I jokingly call foo-foo dust. We have an idea. Can we make some molecules? Can we prove?
Can you invite me to that next time so I can be a part? And foo-foo dust sounds pretty good.
And it is anchored in the chemistry that we have, this ability to make state-of-the-art macromolecules that have these miraculous properties. And so we had designed some chemistry that improved the precision with which you could use endocrine hormones. So if you think about insulin, it's a miraculous substance, but it's a very dangerous substance because it can kill you from hypoglycemia, right? You think about parathyroid hormone, its modulation of phosphate and calcium metabolism, very similar, right? Even these anti-obesity peptides have profound gut effects where many patients can't tolerate them. And everyone goes through this careful, lengthy dose titration to get to steady state. And so the chemistry, what Kent has called precision peptides, is to improve the therapeutic index so that you can be more aggressive with the dosing to get better outcomes and to experience fewer of the side effects. And the first opportunity was in hypoparathyroidism. This is an absence of parathyroid hormone largely driven by surgery to remove the thyroid gland. Para on the sides of the thyroid gland either get damaged or removed in the process, and your calcium levels fall precipitously, right? It's a life-threatening or certainly altering disease. And what we have made is once a week injectable product that functions just as if you were wearing a pump, like a diabetic wears an insulin pump. And this in some ways was a derivative of what we did at Lilly when I had been a part of advancing Forteo for treating osteoporosis. But instead of using PTH for its pharmacological effect as a pulse each day to build bone, now we're using it for physiological replacement, much as a type 1 diabetic would use basal insulin to control their resting, fasting glucose. Now we're doing it for calcium. And so it is that chemistry applied to that disease. It's that chemistry applied to atypical hypoglycemia after gastric bypass surgery. And of course, one looks at, can you use this to make a better obesity product? And there are things that MBX has that are emerging into early development. We'll see how they do. This is a risky business, but we have a reasonable track record.
We talked a little bit about when I asked the question around what can universities be doing, and we talked about retention. I see from time to time an entire team of researchers will move from one university to another. And given the research and the magnitude of success that you've had in research and then translation to new company formation, I'm sure that you've been approached to move from Bloomington to somewhere else. What keeps you in Bloomington? I assume it's not just the 10-0 football team. I assume there's some other things that keep you instead of saying, hey, I'm going to pick up my lab and I'm going to move it to Stanford or wherever that may be. What has kept you tied to so close to Bloomington?
As I said, August of 1974, when I arrived here, I thought I was coming to get an education and I would be gone. And it exemplifies the importance of the university to bring the diversity that we need within the state, right? Here I am, 50 years later, and I've often joked I must have been a Hoosier in an earlier life, or I must be a born-again Hoosier or something of that sort. I love the lifestyle in Indiana. The everyday life is far less stressful than what I experience in New York, California, whether it's the southern part of the state or the northern part of the state. And to be creative, one needs privacy. One needs quiet time to think. And quite frankly, that's been a part of my success in moving to a place like IU relative to being at Lilly, where you're constantly distracted with all of the administrative work, all of the regulatory work, and many would know I've professed that my blood type is HR negative. I just don't deal with all of this, the restrictions that come with let's go while we're young.
That's neat. I like the way you talk about that in terms of needing the space, and I find that to be the case too. Indiana is a wonderful place in that regard, and you found the opportunity in Bloomington to give you that space too, which is wonderful.
With all due respect, as I fly out of Indiana and I look at Indianapolis or I look at Bloomington, I say to myself, "Oh my gosh, we're surrounded by Indiana," And what I mean by that is I worry about the capacity within the state to embrace diversity. It's probably poorly appreciated that at least half of the individuals that have fueled my ability in Bloomington were educated in communist countries. Whether it was Eastern Europe, whether it was Russia, whether it was China, they came to be a part of what I was doing, and I could not have accomplished what I needed to do without that talent. And that is not atypical. This country and the state of Indiana has developed a healthy dependence on foreign talent to fuel its STEM business. And as we go into this period where it feels as if we're being less accommodating, less welcoming to foreign talent, particularly from some of these countries, notably China, I worry who's going to fill that void. Why aren't domestic students embracing this opportunity? Here I am at 72. It's an amazing career, and people are still recruiting my participation with things that are unbelievably exciting. Who wouldn't want to have an opportunity to do that? The psychological income that you get from helping people live a better life. And so somewhere, somehow, we've missed something in explaining why this is such a phenomenal career. As I said, in the early 60s, when we all went off with our slide rules to do what we did. 90% of the folks that entered into graduate school when I went to Bloomington in 74 were domestic. I mean, I treasure the international nature of our business. I'm not speaking against that, but I'm worried that we just don't sufficiently value, we don't sufficiently motivate and recruit talent into the life sciences. And that needs to be corrected from practicing physicians to folks that are doing the basic molecular cell biology, synthetic chemistry.
So it's got two aspects. So we've got to always welcome those outside our borders, and that's been America, and we've got to do better on that front. And then, of course, Americans people who grew up here, interesting them in the sciences, right? And so it's
exactly.
It's both, yeah.
And at this point in time, foreign students, particularly those coming from China, are more questioning, skeptical as to whether they will be welcomed here in the United States. I have no doubt when they're on the campuses of our major universities, there's no challenge. But what when they're out into the rural community? Because I worry about them. Because we're seemingly portraying foreigners as something that is unwelcome and dangerous. We need this talent. And yes, there comes risk with having them there, but we have to find a way to do this, or we're not going to have the capability to advance and to compete.
Before we close, I know you mentioned when we were talking right before we got started today that you're already working on some new ventures. I was going to ask you what comes next. It sounds like you've got some even some new companies that you're thinking about. Is it too early to talk a little bit about what you're working on next?
Can't reveal the specifics of it, but it falls in this heading of my belief that a century or more from now, when people look back at this period and ask what happened, that was so special, that they will look at these biotechnologies, the movement to a more quantitative science, biology now being more mathematical than it ever was. It was more observational, more qualitative. And when I came to Lilly in the early 80s, I always had this sense that this was going to be transformative technology. In 1929, Lindbergh crossed the Atlantic. In 1969, we were on the moon. Look at what that did in that century. Look at what the engineering sciences and telecommunications have meant to us. Does anybody know the chemistry of why we have memory in these devices? Surely, but it's very few folks. And I always asked, in 40 years, where would we be in biotechnology? Could we make the same contribution? And I will tell you, this is the century of biology, as much as the last was a century of physics and engineering sciences. What is the molecular basis of a memory? It's not silicone-based, right? It's not salts, metals that are driving memory. It's DNA, it's protein-based. And so that's what I see coming in the second half of this century. It's unbelievable that we're almost a quarter of the way through it, that we will just have a much better ability to understand physiology, pathology, and intervene with pharmacology. And as I remember my first visits to India, there was always this concern of how you would communicate. How would you lay the cables for communication in India? And then here we are, everybody using wireless communication above. You know, folks just weren't anticipating that. I think what we don't know is going to have the biggest impact on human health. And how that gets applied is more than just the technologists, right? It's the human ethics of it. It's the fairness of distribution and access to these drugs. But we need to do it right. And when I see the screaming for decreasing price on pharmaceuticals, I get it. I understand a drug doesn't work unless a patient has access to it. Right? We have to get access. On the other hand, let's do no harm. The investment community that we have developed is what's leading to these fantastic breakthrough drugs. If we destroy the willingness and the interest of people making an investment, the state of the art will stall exactly where it is right now. And so when I think of what we as technologists are being asked to do, cure cancer, treatments for cognitive dysfunction, for crying out loud, this issue that we look to the politicians to solve isn't as hard. Get us a solution that meets both needs, access and the ability to continue to stimulate technology so that we get ever better drugs.
So you're working on kind of some of those things, it sounds like.
That's where we're going. That
awesome.
is where we're going, and focusing on healthy aging, and even longevity is not off the table.
That's awesome. Can't wait to see what comes next. Looking forward to, well, Dr. DiMarchi, we made it to a more mundane part of the podcast where I ask you some personal questions in terms of your favorite things. So I'll ask you what your favorite movie of all time might be.
That's a challenging one. I suppose Casablanca has always had a special place for my wife and I.
How about your favorite place to vacation?
I think we tend to find the most relaxation and have returned repeatedly is Marco Island in Florida, right, just south of Naples. The sunsets are absolutely spectacular, and the quiet time on the beaches have always been special for my wife and my three daughters.
How about your favorite musical artist?
I think the one that I derive real inspiration from right now is Mick Jagger.
Nice.
His ability just to rock on. You know, he's bringing with him a cardiologist. They have a defibrillator there, and he's just going. It's such an inspiration.
What is the first thing you do in the morning when you get up out of bed?
It's usually a cup of coffee. Typically, when I'm in Bloomington, which I said is more than half my time, I'm first into the lab. And I joke with the guys that I have matured to a point where I only work half days, 7 to 7. and it's far more than I should be doing. But it is so infectious when you've been touched by the magic of these molecules. You see them as chemicals, and all of a sudden they become these molecular miracles that you come in each day with this belief that today may be the day when we have another one of these eureka moments. And I've been asked, what are the most important syntheses of my life? And I've gravitated to responding, my 3 daughters, to which my wife deserves the lion's share of the credit. Family is unbelievably important to me, and my faith in believing that we're incredibly fortunate to be in the place we are, and the reward is the opportunity to do it again, right? Come in each day and have the funding, the people, the laboratory, and I'm very grateful to Indiana University for the support they provide me.
If a young person was coming to you for advice, just generally, somebody who's middle school, high school, in terms of thinking about life, what advice would you give a young person?
Surround themselves with people who have impeccable integrity, that their compass just points north. Bruce Merrifield was one of those guys. Gus Watanabe was one of those guys. Leroy Baker was one of those people for me. And have an oversized ambition for what life is about. It's been said, I think it's Confucius, that we all have 2 lives. And the second one begins when we realize we only have one. And so,
Right.
Being with people that make you appreciate, you're only going to go around once. What is it that when you get to 70 or 80 and look back and say, that's what life was about? I remember telling people when I was leaving New York to come back to Indiana, I am not going here to shuck corn. We are going to do something phenomenal. And I think possibly the biggest contribution this state has ever made to the global community was the success with human insulin, which was a public-private partnership that allowed Lilly to do what it did because it validated that this academic technology that had ended up in this small startup could actually make medicines, and therefore the investment community came rushing to start new companies, and look what it has become. And unfortunately, you don't see enough acknowledgement for what was done there. And I'm not asking for myself. I think Irv Johnson, Neil Pettinger at the top of the organization, but just a host of individuals from Ron Chance, Bill Bromer, Bruce Frank. I mean, I could go on and on. Leroy Baker deserve the recognition for what a phenomenal accomplishment this was that laid the groundwork for people like myself to do what we've done in the last 40 years.
It's amazing. This has been a wonderful opportunity to dive into your background and the successes that you and your teams have had over the years. I just can't thank you enough for spending the time to be on the Indiana 250 podcast. Can't thank you enough. All of us can't thank you enough for the groundbreaking work that affects so many Hoosier lives and, of course, lives around the world. So thank you for all you've done. And here's to a lot more, because I know you're a young 72, so I know there's a lot more coming. So thanks for all you've done.
Thank you so much for the opportunity to speak with you and others. Happy holiday, and to you and all of our listeners. It's a great time. It's a time to be thankful. So I wish you the best.
To learn more about other leaders on IBJ Media's Indiana 250 list, go to indiana250.com and look for a Page 2 feature each week in IBJ. We'll be back with the new Indiana 250 off-the-record conversation soon.